So good morning students. Today in the last class we had discussed about the pistil. Okay, we were discussing about the female reproductive structure. In that I had told you about the pistil. Okay, pistil, it has the three main regions, that is stigma, style and ovary. Okay, this is the stigma, style and one more, that is the ovary. Stigma, which is a knob-like, in some plants it will be pointed shape, it is sticky surface or it is a landing platform for the pollen grains. Style, which is a slender stall or a slender tube-like structure, okay, which ends or terminates into a basal region, that is the ovary. In the ovary, there will be presence of an ovarian cavity. What it is present? That is the ovarian cavity. And this ovarian cavity are known as the locules. Okay. Here in this ovary, there will be a ridge of tissues, and that ridge of tissues are known as the placenta. What it is known as? It is known as the placenta, and here the placenta is going to attach the ovary with the ovules. Inside that there will be presence of what? Ovules. Okay. Except that we had studied about the two terms. That is the one is the fin carpus, and one more, that is the apocarpus. Fin carpus and apocarpus. What do you mean by this fin carpus and apocarpus means, where see carpels, this is regarding the carpels. If carpels are fused, okay, in some flowers the carpels will be freely arranged, okay, and in some flowers freely arranged means how means, it will be like this. These are the carpels which are freely arranged, okay, and if it is freely arranged, then it is known as the apocarpus. Okay, if the carpels are totally fused like this, in this way it is there, so then it is known as the syncarpus. This is very important terms for 1 mark in your theory exam. What is apocarpus? What is syncarpus? Apocarpus means where the carpels are freely arranged. Syncarpus means where the carpels are fused in what we call it is in a fused form. So these all things we had studied. See, if one carpel is there in a flower, then it is known as the monocarpellary. Okay. If two carpels are there, then it is known as the bicarpellary. If it is three, then tri. Many are there means that will be poly or multicarpellary. Except this, we also studied about the number of ovules in a different plants or different flowering plants. If it is one means, one example is there, mango, wheat, paddy. I had told you along with the examples, in some of the flowering plants it will be many. For example, that is watermelon is there, brinjal is there, then thousands will be there. Okay, thousands means it will become in one plant, specifically in one plant, that is the poppy plant. Okay, million, more than million means that is in the orchids. These questions, these points are very important. Keep in mind. Then next we will go for the next structure of the female gametophyte, that is the megasporangium. What it is? Megasporangium. Megasporangium, which is also known as the ovule. Megasporangium. See here, Megasporogenesis, it is also known as the ovule. ovule, which is a very small structure which is present in the ovary. Okay, so here megasporangium, from where it is going to arise means, I told you this is the ovule. Okay, this ovarian cavity is known as the locule. Okay, then here inside this ovarian cavity or in the locules, there will be presence of a ridge of tissues. These are the tissues. Okay, these tissues are known as the placenta. What it is known as? It is known as the placenta. From this placenta only, the ovules are going to arise. Like this. Okay, see here, ovules arises from the placenta. Okay, placenta, which is the ridge of tissues. Okay, so here the ovule is a small structure attached to the placenta. So let's see the structure of an ovule. Here, the structure of ovule. This is one of the very important question for your theory exam. That here I will write structure of ovule, or else they will give explain the structure of ovular megasporangium, or sometimes only they will ask draw a neat label diagram of ovule. That much only. So let's see here the diagram of ovule. This is the question for 5 marks. Either they will ask the structure of ovule, or either they will ask the diagram of ovule. Describe the structure or diagram of ovule. So let's see here. Now see here, this is ovule. It is one of the small structure attached to the placenta. What is ovule? This is one of the small structure attached to the placenta. Okay, and which structure is going to attach this to the placenta means by means of a stall. By means of a stall, and that stall is known as the funicle. What it is? That is the funicle. See here, this is the ovule, the structure of the ovule or main body of the ovule, where this region is labeled as the funicle. Okay, here a stall is there which is attached to the placenta. Okay, about placenta I have told you. This region, the main body of the ovule, is attached to the placenta by a stall. Okay, this stall is known as the funicle. What it is known as? It is known as the funicle. Then here, that particular stall, that is the funicle. Here, the body of ovule. This is the main body of the ovule. The body of ovule attached with the funicle by a structure. This structure is known as, here this is the small depression, what you can see, this structure is known as the hilum. What it is known as? It is known as the hilum. See, funicle is a stall which is attaching the placenta with the ovule. Okay, and here the main body of the ovule attaches with the funicle. Here you can see, this is the main body. This is the funicle. Okay, the funicle is attached to the main body by a structure or a region. This region, it is known as the hilum. What it is known as? Hilum. So hilum represents the junction between the funicle and the ovule. Here I write hilum is the junction between funicle and ovule. Okay, here the hilum is going to attach the funicle with the what we call ovule. So this and here it arises at two envelopes. Okay, or two coverings. What are the two coverings? You can see this is one covering of the ovule, outer covering, and this is the inner covering. So these two envelopes or these two coverings are known as the indicuments. What it is known as? It is known as the indicuments. Here this is the outer indigument and one more envelope is there. This is the inner indigument. Okay, there are two envelopes in these ovule or the two coverings which are known as the outer indigument and the inner indigument, which is enclosing the ovule body. Okay, how many indiguments? That is the two indiguments are there. And see here, these two indiguments are encircling the ovule or enveloping the ovule, not completely. At the tip region, it is going to leave some space. Okay, and our whole-like structure it is going to leave at what? At the upper region of the ovule, and this region is known as the microphylar region. What it is? It is the microphylar region. Okay, here, listen here, integument encircle the ovule. not completely. At the tip of the ovule, here at the tip of ovule, a small opening will be there, and that opening is known as the micropyle opening, or that region. This opening, you can call it as the micropyle, and that region is known as, the tip region is known as the micropylar region. See here, at the tip of ovule, indicuments, two indicuments forms a hole or a pore. That hole or pore is known as the micropyle, and that region is known as micropylar region. What is that region is known as? That region is known as the micropylar region. Okay, then opposite to micropylar region, where one more region is there, which is totally opposite to the micropylar region, and that region is known as the chalazal region, or else you can also call it as the chalazal end, where the two integuments comes and meet. Okay, representing the chalazal end represents the basal region of the ovule. See here, I will draw this one. Next, chalazal end. Okay, chalazal end is an opposite region to the micropylar region. It lies, or it is a opposite region of micropylar region, and it represents the basal region of an ovule. You can see the micropylar region is present at the tip of the ovule. Chalazal region, which is present at the base of the ovule. Okay, what is chalazal? That is it representing the basal part of the ovule. Okay, here whatever the two integuments are, you can see outer integument is enclosing the ovule, and the inner integument, this integument, is enclosing a structure. What is that structure means? The structure is known as the nucleus. What it is? That is the nucleus. Here, integuments enclosing a mass of cells are a part which is known as the nucleus. Okay, here you can see the red colored cells. What you can see, red colored cells, you can see that cells of nucleus here you see that cells, whatever the red colored cells are there, these are the cells of nucleus. These cells of nucleus have an abundant reserve food material. What does it have? It has an abundant reserve food material. Okay, the cells, what you can see here in this structure or in the nucleus, that cells has a very large amount of reserve food material. Okay, so here at the center of this nucleus there is a presence of a female gametophyte. What is located over here? That it is the female gametophyte, or else you can also call it as the embryo sac. What it is? That is the embryo sac, or which is also known as the female gametophyte. This structure, a blue color structure in which there is a presence of certain cells, that is known as the embryo sac or female gametophyte. Okay, and ovule generally it has a single embryo sac, okay, which is going to formed from a megaspore mother cell. This embryo sac is formed from the only single embryo sac to be located in each ovule, and this embryo sac is formed from the megaspore mother cell. That we can also write it as the MMC. Okay, megaspore mother cell. And the thing is that next, how this embryo sac or how this female gametophyte is going to form. You can see the structure over here. This is the female gametophyte. How it is going to form, that is very necessary. That is very important. So we will study in the next topic, that is known as the megasporogenesis. In the male gametophyte or in the male reproductive structure, you had studied about the microsporogenesis. That is the process of formation of pollen grains from the pollen mother cells. Okay, here in this megasporogenesis, that is the process of formation of megaspore, or else you can say embryo sac from the megaspore mother cells. So let's see about that process. And this diagram, sometimes they will ask to describe the structure of an ovule, sometimes they will ask only the diagram. This diagram is very important for your theory exam. For 5 marks it will be asked. Okay, mark it as very important question for your exam. The next term we will go for the process how this megaspore is going to form. Next term, that is the megasporogenesis. Okay, here what do you mean by megasporogenesis? Means the process of formation of megaspore from the megaspore mother cells. Okay, from the megaspore mother cell, MMC. Okay, so this is known as this process is known as where there is a process of formation of megaspores from the megaspore mother cell. Okay, this process is known as the megasporogenesis. See, one ovule will develop one single megaspore mother cell. Okay, one ovule generally differentiate a single ovule generally differentiate a single megaspore mother cell. Okay, in which region means where it is going to develop a megaspore mother cell, or where the megaspore mother cell is going to differentiate in the ovule means in micropylar region. I think you know what do you mean by this micropylar region of what? Of nucleus. Okay, by the ovule, and that cell we can consider it as the megaspore mother cell. So that cell I am going to draw over here. Let's see about that one. This is the cell which is having a dense cytoplasm. This is the dense cytoplasm, and this is one larger cell which is having a dense cytoplasm and a prominent nucleus. Okay, it will be like this. Next, here around this is located where, or it is differentiated where, is that is at the micropylar end or micropylar region. This is the micropylar region of an ovule where this larger cell is present, and here around this cell there will be presence of a nucleus, another cells. Like this. Okay, this is the larger cell with the dense cytoplasm and a prominent nucleus. This cell is known as the megaspore mother cell. Okay, it is known as the megaspore mother cell, and this cell is the nucleus. Understood? Okay, now next this megaspore mother cell will undergo into a division, and that division is known as the meiotic division. It will undergo into meiotic division. That is the reductional division. Which cell? This cell. This cell will undergo into the meiotic division. After undergoing into the meiotic division, first it will divide into the meiotic 1 division and into the meiotic 2 division, where it will give rise to the 4 megaspore cells. So let's see here. I will draw this one. You see, This cell is undergoing into the meiotic first division, where this cell will divide into two cells. Okay, this one megaspore mother cell has undergone into the meiotic division. It will form the two cells. Okay, with the cytoplasm and with the nucleus. And the other nuclear cells will be like that only. They are not going to divide. Okay, like this. And this is the micropylar end, as you had seen. This is the, and if one megaspore mother cell divided into two cells, then we can call it as the megaspore dyad. Dyad means two-celled stage. Okay, my megaspore dyad. The next again, and here first division, meiotic division they had undergone, and this is the meiotic. This is a meiotic one division, and this is a meiotic second division. Again, these cells will undergo to the meiotic division. Here you see, if it is undergoing again into the meiotic division, it will form the four cells. How many cells? That it is four cells. Like this. And the remaining, the nuclear cells will be like that only. They will not undergo into any division. Like this. And this stage, you can see 1, 2, 3, 4, and you can call it as the megaspore tetrad. What it is called as the? It is called as the megaspore tetrad. Listen here carefully. What happens means, here in the ovule, in this micropylar region, okay, in this micropylar region, this is the micropylar region where there is a presence of nucleus. Okay, inside this nucleus at the micropylar region, only one cell starts to differentiate. Okay, that one cell I had shown over here, that cell is the larger cell which is having a dense cytoplasm and a prominent nucleus. So this cell is the megaspore mother cell. Already I told you one ovule will give rise to or differentiate only a single megaspore mother cell. But that megaspore mother cell will undergo into the repeated division. So here this megaspore mother cell will undergo into the meiotic division, first division. Here it is going to divide the two cells. That two cells we can call it as the megaspore dyad, means two megaspore cells. These will undergo into the meiotic two division, resulting in the formation of four cells, and that is known as the megaspore tetrad. Okay. Further, what happens here? Let's see. So this is about the production of four cells. Keep in mind. Okay, one megaspore mother cell has given rise to the four megaspore mother cells. Next, we will go for the female gametophyte. How the embryo sac is going to form from this megaspore. Now see here. Next, that is the female gametophyte. Here, in the female gametophyte, in majority of flowering plants, one of the megaspore is functional. Okay, among 4 megaspore, see here, these are the 4 megaspore, what you had studied in the last topic. Among these 4 cells, only one will be the functional in majority of angiosperms. See here, in majority of flowering plants, or angiosperms, among 4 megaspores, only one will be the functional. Will be functional. How many will be the functional? That is, only one will be the functional. So this one functional megaspore, here you see one functional megaspore will give rise to the, or from this one functional megaspore, one embryo sac is going to develop. One embryo sac will develop. From how many? One megaspore, only one embryo sac is going to develop. Okay, and this method of formation of one embryo sac from one megaspore, this development is known as the monospermic development. Altogether, this development is known as monospermic development. Mono means single. Okay, from single megaspore, a single embryo sac is going to form. Sorry, monosporic is monosporic development. Sometimes they will ask for 1 mark. Simple question for 1 mark, that is development of one embryo sac from the one megaspore. Okay, that is known as the monosporic development. So next we will see about the how the embryo sac is going to form. Next, female gametophyte or else formation of embryo sac of female gametophyte. Okay, in this formation, here see, already you had studied about there is a formation of 4 cells like this. 1, 2, 3, 4. Okay, so this we are calling it as the megaspore tetrad. What it is? That it is the megaspore tetrad. Okay, among these 4 cells, only one will be the functional. What I told. Okay, here if for example this is the functional. Okay, this is the functional and remaining 3 will degenerate in this way. Okay, remaining 3 will degenerate. Only one cell will be the functional cell. Okay, so these cell will divide mitotically. This is the megaspore cell. This megaspore cell will divide mitotically, not meiotically, mitotically. How it is going to divide? This one functional cell will divide. This is the functional megaspore cell will divide mitotically. If it is going to divide mitotically, one cell will give rise to the 2 cells. I will show you here. For example, this is the one cell. Okay, one structure or the embryo sac where 2 cells are going to divide. Here you see this one cell will get divided into 2 cells. These are the 2 cells, and then 2 cells will divide and they will move at the opposite sides or the opposite poles. Okay, and this stage we can call it as the 2 nucleate stage. Means where there is an, see, one more thing, in a cell, in this cell only nucleus is going to divide. Okay, so that's why we are calling it as the 2 nucleate stage or 2 nucleate cell also, 2 nucleate stage of embryo sac. Okay, where the 2 cells are divided. Then again these 2 cells, again it will undergo into the mitosis. Okay, if it is undergoing into the mitosis, this will result in the formation of 4 cells. 2 cells will be at the one opposite side, one opposite pole, and the another 2 cells will be at the another opposite poles. And this stage of embryo sac is known as the 4 nucleate stage of embryo sac. Okay, again one more time, these 4 cells again it will undergo into the mitosis division, which will give rise to the how many cells? 1 cell mitotically divided to give 2 cells. 2 cells divides to give 4 cells. If 4 cells are going to divide, how many cells are going to form? That is 8 cells are going to form. These are the 4 cells at one side, one pole, and 4 cells at another pole. And this stage we can call it as the 8 nucleate stage of embryo sac. What it is known as 8 nucleate stage of embryo sac. Okay, so how many nucleus you can see over here? That is 8 nucleus you can see over here. Okay, so here strictly there is a nuclear division and there is no cell wall formation. That's why only the nucleus is going to divide, not a cell is going to divide. Okay, so here 6 of the 8 cell nuclei are surrounded by cell walls and organized into the cells. How means? I will show you here. After this, see here, 6 nucleus, they are going to form a cell wall, and 2 nucleus, they are not going to form any cell wall. How means? You see here that it will be like this. 6 will arrange at one pole, one region. 2 cells will not have any cell wall, and another 3 cells will have a cell wall. Like this. And here they have these are the 8 cells. What we can see there, 8 cells, they have rearranged themselves by the, when they are going to rearrange, when there is a formation of cell wall. 2 cells are, there is no cell wall formation, but 6 cells has developed their cell wall. Okay, so here you see among these 8 cells, first there is an presence of 3 cells at one side. Which side means that is at the micropolar end. There is a presence of 3 cells. At what end? That is at the micropolar end. Remember the structure of ovule. These 3 cells, it is known as the antipodal cells. Which cells? That is the antipodal cells. Okay. Next, here in the chalazal region, there is a presence of 3 cells, 2 synergids. 2 cells, we can call it as the synergids, and 1 egg cell. Okay, altogether we can call these 2 synergids. See here, 2 synergids and 1 egg cell. And here 3 antipodal cells are there. Okay, altogether we can call these 2 synergids and 1 egg cell as egg apparatus. What we can call? We can call it as the egg apparatus. Then here there is a cell at the center of the embryo sac, and this cell is known as the central cell. What it is known as? It is known as the central cell, and it has the 2 nuclei. That is the 2 polar nuclei. Okay, so this is about what? Embryo sac formation. And here one more thing. In this way, the cells gets distributed in the embryo sac, and the cells are grouped together at the micropolar end as well as at the chalazal end. At the micropolar end, there is a presence of egg apparatus. This region, we can call it as the micropolar end. Okay, where the egg apparatus is located, and this region where the antipodal cells are located, this region is the chalazal region. And whatever the egg apparatus in turn consists of 2 synergids and 1 egg cell. Okay, the synergids has a specialized thickness. What it is? It has some specialized thickness. These thickness are known as the specialized thickness, and these thickness are known as the filiform apparatus. What it is known as? It is known as the filiform apparatus, and this filiform apparatus play a very important role while the pollen tube is going to enter into the embryo sac. Okay, and 3 cells are at the chalazal region, which are known as the antipodal cells, and the central cell, as mentioned earlier only, there is no cell wall formation, and you can see 2 polar nuclei. Okay, so the large central cell, which is having the 2 polar nuclei, and a typical angiospermic embryo sac at maturity, it shows a 8 nucleate, 7 celled stage. 8 nucleate, 7 celled stage. Okay, maturity, a typical or angiospermic or angiosperms embryo sac will show a what? Will show a particular, how many cells are there? 8 nucleus. 8 nucleus means 1 nucleus. You just see the nucleus. 1 nucleus, 2 nucleus, 3 nucleus, 4, 5, 6, 7, 8. 8 nucleus are there. 1, 2, 3, 4, 5, 6, 7, 8. Okay, count the cells. 1, 2, 3. This together, we cannot consider it as the 4 and 5. This together is the 4 because there is no cell wall formation. This is the 5, 6, 7. 8 nucleus are there, but 7 cells are there. Okay, this stage is known as the 8 nucleate 7 cell stage. So this question is also very important. Draw a neat label diagram of mature embryo sac. Now I am going to draw it. This is the last topic for today's class. Only diagram is there. Here you see, this question will be asked for 3 marks for your theory exam. What it is? That is the mature embryo sac. Draw a label diagram of mature embryo sac. How it will be means, sir? See here. Specialized thickening will be there. This thickening is known as the filiform apparatus. If they ask for 3 marks, draw a label diagram of mature embryo sac. This diagram you have to draw it. Okay, these are the 2 synergids. One egg cell, central cell, and here is the polar nuclei. Next to that, these are the antipodal cells. Okay, so this is about what? A mature embryo sac. Okay, tomorrow I will go with the next topic, that is the pollination. Today's assignment is there, that is describe the structure of anatropous ovule. One more, draw a label diagram of embryo sac. Okay, these are the two questions for your today's assignment. Please just put it in your WhatsApp group. Okay, don't do any cheatings over here. Okay, if you are okay, thank you.